part 3. In today's lecture, we'll be discussing about the anatomy of SARS-CoV-2. Since this virus targets and infects the lung, so and therefore we will be discussing about the anatomy of the lung as well. Since anatomy, pathogenesis, pathophysiology of SARS-CoV-2 is similar to COVID-19. So when we discuss about the anatomy, pathogenesis, pathophysiology of SARS-CoV-2, it means we discuss about the anatomy, pathogenesis and pathophysiology of COVID-19, because both viruses have the same gene, same nucleic acid and same RNA. In 80 to 85% of cases, both viruses have got the same gene, same RNA and same nucleic acid. So when we discuss about the anatomy of COVID-2, it means we discuss about the anatomy of COVID-19. So from outer surface to inner surface of this virus, we will see these following structures. First structure that we see, this is called membranal protein. What protein? Membranal protein. Over the surface of this virus, we see a special protein which is called membranal proteins. After this membranal protein, we have double lipid layers. After this lipid layer, we have got capsid, and within capsid we have got nuclear capsid. Let's move again to membranal protein. Within this membranal protein, we see different proteins with different sizes, with different shapes, functions, and structures, which could be explained here one by one. The first one is membranal protein. For short, they say, or M protein. The second one is called S protein, spike protein or S protein. Another protein is called envelope or E protein. Another very important which is protein, which is present into the inner surface of this virus, that is called nuclear protein. So over the surface of this membranal protein, we have got three proteins: membranal protein, another is spike protein, membranal protein, envelope protein. Spike protein we use for spike protein S, for membrane protein we use N, and for envelope protein E. Remember one another protein which is present into the inner surface of this virus, that is called nuclear protein. Within this capsid in which the nuclear protein is located, it is bound to positive single stranded of RNA. So from outer surface to inner surface of this virus, we see membranal protein. After the membranal protein, we see lipid layer. Within the lipid layer, we see capsid. Within the capsid, we see nuclear capsid. Within the nuclear capsid, we have got nucleic acid, which is bound to positive single strand of RNA. Let's start from spike protein. This spike protein has a major role in the shape of the virus, or anatomy of the virus, pathogenesis or pathophysiology of the virus, treatment of the virus, as well as designing of the vaccine. So S protein, spike protein or S protein has a major role in anatomy or shape of the virus, pathogenesis or pathophysiology of the virus, and treatment of the virus, as well as designing of the vaccine is related to the spike proteins. This spike protein is made from two parts. One is part 1, another is part 2. This part 1 of protein binds to a special receptor which is called S2 receptor. This spike protein is made from two parts, S1 and S2. S1 binds to spike protein, to S2 receptors, and the S1 and the S2 helps the virus to enter or to diffuse into the cell membrane of the virus. So spike protein is made from two parts, S1 and S2. S1 is bind to S2 receptor, and S1 helps the virus, or S2 helps the virus to diffuse into the cell membrane, and from cell membrane this virus is entering into the host cytoplasm. The process by which this virus is entering into the inside of the cell, that is called endocytosis. So let's have a few words about the S receptor, or angiotensin converting enzyme receptor 2. This S2 receptor is present over surface of pneumocyte type 2. In addition, this virus which is present over the pneumocyte type 2, this virus is also expressed in the lungs, heart, intestine, as well as kidneys. So remember the affinity of this virus for capturing of the virus 25 times more than the virus. So and therefore this virus rapidly and fastly spreads to the body and produce or lead to severe clinical or critical manifestation of the patients. This was about the S2 receptor. First was spike protein. We have discussed about the spike protein is made from two parts, S1 and S2. S1 binds to S2 receptor. S1 helps the virus to enter to the membrane of the cells, and finally this virus through this S2 receptor is entering into the host cytoplasm. S2 receptor is a receptor which has got a special affinity for capturing of the virus. So and therefore this virus rapidly, fastly spreads to the body and lead to severe or critical manifestation. This was about the spike protein. Spike protein has a major role in anatomy, shape, pathogenesis, pathophysiology, treatment, designing of the vaccine, even pathogenesis of the virus, even rapid spread of the virus, even severe or clinical manifestation of COVID-19 or COVID-2 is related to this is spike protein. After the spike protein, we have got another protein which is called membranal proteins or M protein. This membranal protein, the large number of this M protein are present over the surface of the protein or within the membranal proteins. And this M protein has envelope shape. It has generally three main functions. M protein has got three main functions. The first one, this virus has a major role in organizing of assembly of the virus, as well as an interaction between the virus and cellular protein. Generally, this M protein has a major role in replication of the virus. So the first protein was the spike protein, the second protein was M protein. The large number of this M protein are present over surface of this viruses. This M protein has three major functions. The first function of this M protein is organizing of assembly of the virus, as well as an interaction between the virus and cellular protein. Generally, this virus has a major role in replication of the virus. This was the function of the M protein or membranal protein. We have already discussed about the S protein, which is also present over surface of the virus, and the second protein that we have discussed already, that was the M protein. The third protein, which is present over the surface of the membranal protein, that is called E protein or envelope protein. E stands for envelope protein. This protein directs and guides the virus towards the cellular workshop or intracellular machinery or intracellular manufacture, generally in the plasmic reticulum as well as Golgi or Golgi apparatus. In short, this E protein directs and guides the virus towards the endoplasmic reticulum and Golgi apparatus. This E protein, like M protein, has a major role in replication of the virus. Remember, let's go to the another protein. This protein which is present into the inner side. These three other protein were present over the surface of the cells. The last one, it is presented in the inner side of the virus. Present in the inner side of the virus. This nuclear protein has a crucial role or a definite role in inhibition of the immune systems of the cells. After inhibition of the immune system of the cells, this virus leads to cellular injury. After the cellular injury, it leads to apoptosis, and finally cell membrane is destroyed, and the cell membrane is dries, and the virus spreads to the all body. This was the nuclear protein. This nuclear protein has a major role in inhibition of the, for sure, this N protein, which is located into the inner side of the virus. It has a major role in inhibition of the immune system, cellular injury, finally death of the cell of lungs. And then the virus rapidly and fastly spreads to the throughout of the lungs. This was about the nuclear protein. Generally, this virus is made from 4 proteins. 3 of them are located over the surface, that is called surface or membranal protein. Another one which inhibits the immune system, and this is called nucleic acid or a nuclear protein, which is bound to positive single strand of RNA. It inhibits immune system, lead to cellular injury, finally apoptosis and death of the cell. This was about those important protein which has a major role in replication of the virus, pathogenesis of the virus, pathophysiology of the virus, treatment of the virus, as well as designing of the vaccine. This was about the membranous protein. After the membranous protein, we have got another layer which is called lipid layer. This was membranous protein. The second was lipid layer. This lipid layer is very sensitive to soap. So and therefore, hand washing is one of the most important steps in prevention of this virus. So by using of soap, this lipid layer is dissolved and virus escapes. So first was membranous protein. Under the membranous protein, we have got lipid layer. Below the lipid layer or beneath the lipid layer, we have got another layer which is called capsid. Within the capsid, we have got another layer which is called nuclear capsid. With this nuclear capsid, this nuclear capsid contains genetic materials, and it has a crucial role in pathogenesis of the COVID-19. So, how many layers we have got for this virus? This virus from outer to surface is made from 4 layers. The first one, membranous protein. This membranous protein is made from different proteins with different sizes, different shapes, and different functions, and different structures. We have got over the surface of this virus 3 major proteins. First one is spike protein, the second one is membranous protein, and the third one is envelope protein. Remember, you should not be confused about the nuclear protein. This nuclear protein or this nucleic acid, it is not present over the surface of the protein. It is present into the inner surface protein, and it is bound to positive single strand of RNA. We have got nucleic acid, and nucleic acid is bound to positive single strand of RNA. After this membranous protein, we have got lipid layer. What is the clinical role of lipid layer from the standpoint of prevention? This lipid layer is very sensitive to soap. So and therefore, hand washing is one of the most important steps for prevention of the virus. By using of soap, this lipid layer is dissolved and the virus is killed. This was about the lipid layer. And we have got under the lipid layer the capsid. Within the capsid we have got nuclear capsid. Within the nuclear capsid we have got nucleic acid, which is bound to positive single strand of RNA, and it has a crucial role in inhibition of immune systems. After inhibition, cellular injury. After cellular injury, apoptosis. After the apoptosis, the cell is dead. This was about those proteins and those structures that we have mentioned about the SARS-CoV-2. Why we discuss about SARS-CoV-2? Because anatomy, pathogenesis, pathophysiology, and pathophysiology of both viruses, COVID-19 and COVID-2, are similar. So we discuss about the anatomy of CoV-2. But actually, this COVID-19 is also anatomically similar to COVID-19. So when we discuss about the anatomy of CoV-2, it means we discuss about the anatomy of COVID-19, because both viruses have the same gene, same nucleic acid, and same RNA. That is why when we discuss about COVID-2, it means we discuss about the COVID-19. Since this virus targets and infects the lung, so and therefore we'll be discussing about the anatomy of lung. We have got a brief overview about the anatomy of the lung, and we'll be discussing about the important anatomical structures of the lung. Since respiratory system is divided into 2 parts, upper respiratory tract and lower respiratory tract. Upper respiratory tract, we start from the larynx. This is the larynx. Within the larynx, we have got vocal cord. Below the vocal cord, we have got thyroid ligament. This thyroid ligament is a very important place because of tracheostomy. When we want to do tracheostomy, we should use the thyroid ligament. After the thyroid ligament, we have got cricoid. Cricoid or Adam's apple. Along the cricoid, we have got trachea or windpipe. This trachea or windpipe is supported by incomplete C cartilage to keep the trachea open or patent. This is the function of the incomplete C cartilage. Through this trachea, oxygen is coming from atmosphere into the lungs, and carbon dioxide through this trachea goes to out. Trachea. Right now, this trachea is immediately divided into left main bronchi and right main bronchi. Left and right main bronchi. This left and main bronchi is also supported by incomplete C cartilage. Right now, this left main bronchi again divided into the 2 lower bronchi, and the right one is divided into 3 lower bronchi. Finally, these lower bronchi are divided into segmental bronchi, and segmental bronchi finally is ended by terminal respiratory bronchioles, and terminal respiratory bronchiole finally is divided as ended by the alveoli. Within the lung, we have got more than 600 millions of the alveoli. Within this alveoli, oxygen gas exchange takes place. Oxygen we breathe in, carbon dioxide we breathe out. Generally, these alveoli are responsible for gas exchange between breathing air as well as pulmonary circulations. The pulmonary circulation. This pneumocyte have got to is made from 2 types of the cells: pneumocyte type 1 and pneumocyte type 2. Pneumocyte type 1 is responsible for gas exchange, and pneumocyte type 2 is responsible for creation of surfactant. What is surfactant? Is a chemical substance which prevents collapsing of alveoli by reducing of surface tension of the pressure, surface tension of the alveoli. This was about the function of the alveoli. What else? Another. Again, we will repeat rapidly from top to bottom. In today's lecture, we will be discussing about the anatomy of SARS-CoV-2. Since this virus targets and invades the lung soon, therefore, we will have a brief overview about the anatomy of the lung. First, we will start from anatomy of the SARS-CoV-2. Since anatomy, pathogenesis, pathophysiology of SARS-CoV-2 is similar to COVID-19. When we discuss about the anatomy of COVID-19, it means we discuss about the anatomy of COVID-2 because both viruses have got the same gene, same nucleic acid and same RNA. In 80 to 85%, both viruses have is made from same gene, same RNA. So from outer surface to inner surface of this virus, we will see the following anatomical structure. First, the membranal proteins. After the membranal protein, we have got lipid layer. Within the lipid layer, we have got capsid. Within the capsid, we have got nuclear capsid. Within this nuclear capsid, genetic material is present. Right now, we will be discussing about the membranal protein. These membranal protein have got different sizes, different shapes, different function, and different structure. The first one, which is very important, is the spike protein. This spike protein is made from 2 parts. Part 1 or part 2, S1 and S2. For spike protein, we also use for short S. This S protein is made from 2 parts, S1 and S2. S1 binds to the S2 receptor, and S2 helps the virus to diffuse into the cell membrane. Finally, this virus, through a special process which is called endocytosis, is entering into the host cytoplasms. This was about the membranal proteins and spike protein. And spike protein and S2 receptor should not be forgotten, because spike protein has got a special affinity for capturing of S2 receptor. And remember, this S2 receptor also is interested to capture the virus. Both of them together increase the rapid spread of the virus to the body. This was about this S2 receptor is not just limited to the lungs. It's also present in the lungs, hearts, intestine, and kidneys. This was about the angiotensin converting enzyme receptor. After the spike protein, membranal protein. What is the major role of membranal protein? Generally, it has got a major function in the replication of the virus. The second thing is envelope protein or E protein. This envelope directs and guides the virus towards the endoplasmic reticulum as well as Golgi apparatus. After the E protein, we have got another protein which is called nuclear protein. This nuclear protein is not present over the surface of the virus. This protein is present into the inner surface of the virus. Inner surface of the virus. And this protein has a major role in inhibition of the immune system of the virus. So after the membranal protein, this was the membranal protein, and this was the lipid layer, and this was the capsid, and the another one is nuclear capsid. Within this nuclear capsid, we have got genetic material which inhibits the immune system and lead to cellular injury and finally death of the cells. This was about the anatomy of SARS-CoV-2. Let me again discuss about the anatomy of the lungs. Lungs divided into 2 parts, upper and lower respiratory part. Upper, we start from the larynx. Within the larynx, we have got chorda tendineae. Below the larynx, I have got thyroid ligaments. This thyroid ligament is a very good place for tracheostomy. Below the thyroid ligament, we have got cricoids. Cricoids are also called Adam's apple. Along the cricoid, we have got trachea. This trachea is supported by incomplete C cartilage to keep the trachea open or patent to oxygen goes through this trachea, goes out, and carbon dioxide goes out, and carbon dioxide, oxygen is coming in. After this trachea immediately is divided into two parts, left main bronchi and right main bronchi. Left main bronchi is divided into two. Left one is divided into two lower bronchi. Right one is divided into three lower bronchi. Lower bronchi divides into segmental bronchi. Segmental bronchi finally produces or makes terminal respiratory bronchiole. Finally, terminal respiratory bronchiole is ended by the alveoli. Alveoli is a place we have got more than 600 million of alveoli. Within the alveoli, gas, carbon dioxide, oxygen, gas exchange takes place. We breathe in oxygen, we breathe out carbon dioxide. Generally, these alveoli are responsible for gas exchange between breathing air as well as pulmonary capillary circulations. We have got two types of pneumocyte within the alveoli. One of them is called pneumocyte type 1. The another one is called pneumocyte type 2. Pneumocyte type 1 is responsible for gas oxygen, and pneumocyte type 2 is responsible for creation of surfactant. What is surfactant? A chemical substance which prevents collapsing of the alveoli by reducing the surface tension of the alveoli. It keeps that the alveoli is not collapsing. This was about the anatomy of the SARS-CoV-2. Thank you so much for your listen. Now, tell me about the anatomy of the coronavirus.